Hi all, welcome to our educational video on the steps in the cell cycle. I am OLOGY. In this video, we will explore the fascinating process by which cells reproduce and grow. Let's dive in. The cell cycle is an essential process that allows living organisms to grow, develop, and repair damaged tissues. It is a tightly regulated sequence of events that cells go through to replicate themselves. In eukaryotic cells, or cells with a nucleus, the stages of the cell cycle are divided into two major phases, interphase and the mitotic M phase. During interphase, the cell grows and makes a copy of its DNA. During the mitotic phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. Interphase. During interphase, the cell prepares for division by carrying out various processes. It consists of three distinct stages, G1, S, and G2 phases. During G1 phase, also called the first gap phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. Next comes the S phase, where DNA synthesis occurs. During this stage, the cell carefully duplicates its DNA to ensure that each new daughter cell receives an identical copy of the genetic material. It also duplicates a microtubule organizing structure called the centrosome. The centrosomes help separate DNA during M phase. During the second gap phase, or G2 phase, the cell continues to grow and prepare for the next phase. It synthesizes proteins necessary for cell division and checks for errors in DNA replication. The G1, S, and G2 phases together are known as interphase. The prefix inter means between, reflecting that interphase takes place between one mitotic phase and the next. Mitosis. After interphase, the cell enters mitosis, the phase where division actually occurs. During the mitotic phase, the cell divides its copied DNA and cytoplasm to make two new cells. M phase involves two distinct division-related processes, mitosis and cytokinesis. Mitosis can be further divided into four stages: prophase, metaphase, anaphase, and telophase. During prophase, the cell condenses its replicated chromosomes, prepares the spindle apparatus, and nuclear envelope starts to break down. In metaphase, the chromosomes align at the center of the cell, forming what is known as the metaphase plate. Spindle fibers attach to each chromosome, ensuring the subsequent separation. Anaphase is characterized by the separation of sister chromatids. The spindle fibers shorten, pulling the chromatids towards opposite poles of the cell. Finally, during telophase, nuclear envelopes reform, and the chromosomes decondense. Two identical nuclei form, setting the stage for cell division to complete. In cytokinesis, the cytoplasm of the cell is split in two, making two new cells. Cytokinesis usually begins just as mitosis is ending, with a little overlap. Importantly, cytokinesis takes place differently in animal and plant cells. And that concludes our journey through the steps of the cell cycle. From interphase to mitosis, each stage plays a vital role in ensuring the accurate reproduction and growth of cells. By understanding these processes, scientists can explore various aspects of life, from embryonic development to cancer research. Thank you for joining us. Don't forget to like, share, and subscribe to our channel for more fascinating educational videos. Until next time.